Okay, so the requirement for this question is disagreement. So you've got to pick a side and fight fervently for it. But here's the question. Being able to diagnose a relapse has gotten much better. We've got much more sophisticated tests. So I think we're, at least in my world of watching friends that have had children that have relapsed, we're catching them earlier, it seems, where we're dealing with these single-site relapses and that type of thing. So when I'm telling the story that I'm about to tell, that's the type of patient that I'm talking about and we're arguing fervently about. You know, going through, when we get diagnosed with high-risk disease, we get to go in this knowing whether a high-dose strategy or an intermediate dose strategy or a low-dose strategy would do. Because we've had phase 3 trials to see what is going to produce remission, more likely for more kids. So we know, for example, that you know, coming out of 3891, that in that situation, transplant was better than continuing chemo. In relapse, we don't have that. What we get to compare is a response rate. So taking the Cytoxan has this response rate, and these risks, you know, et cetera. But in these kids that have these single-site relapses, how do we as parents make decisions between going for, you know, kind of a high dose, throw everything at it, try and get back to remission really quickly, kind of following the same strategy that we did during induction, versus going with something that's a low-dose strategy, like a low-dose retinoid or an ABT-751 or 7701, or just doing, you know, acting. How do we make that? How do we make that decision? What things do we need to think about? And we can't talk about quality of life. Because we trust us. We know quality of life.
Well, Mark, I don't think it's fair to say that question and then say we can't talk about quality of life. Because when, I mean, I just don't, because that's excluding a major factor of how parents and physicians make decisions. If quality of life didn't matter, we could do all kinds of things. I mean, there's all kinds of treatments and places we could send people. But when you're faced with a disease that you have a small, if any, chance of actually curing, but you're hoping to get long-term quality of life, or long-term stability of disease, you have to take into account quality of life. And the child is certainly taking it into account, even if the parents aren't. I mean, what the child does and thinks about his treatment regimen impacts how the child is going to respond to that regimen. So, from my own perspective, if I had a child who had a single lesion site of, let's just make it over to make it easier, I would take into account whether that child already had a central line or didn't have a central line, whether that child was symptomatic or not symptomatic, whether they were in school or too young for school, when what their counts were at the time and what they weren't, and what the parents wanted to do. I mean, I wouldn't put a child who was 2 years old on an oral drug if the parents have trouble getting Bactrim into them, you know. But I wouldn't put a 7-year-old on an IV drug if going to school every day is really important. I don't know which one's going to work, so I have to take into account quality of life and what the family thinks is important. And I'm not the judge, let me make it clear, of what that quality of life is for that patient. The family is the judge. I've had patients say, no, really, truly, I don't want an oral drug because I get too stressed making sure that the dose is okay for my child. I'd really rather have the IV because then it's your responsibility. I've had other families just flat out refuse to come to the clinic and we have stuck with oral drugs. So the decision has to be in line with what is right for that patient, and the patient has to guide me in telling me what they need and want. And if we try one thing and it doesn't work, then that gives us guidance as to what to do next. It doesn't tell us, okay, well, we gave ABT-751 and that didn't work, so that means I have to go down this route. They may say, gosh, I really couldn't stand taking an oral medication and I want an IV next. There's so many factors that I just don't think.
Well, I agree with you. My point wasn't to say let's just throw quality of life out the window. I guess what I'm trying to do is come up and see if there's a formula. Is there more, are we more, if we've got a generally healthy person with relapses in a single site, are we more likely to achieve a remission by doing a really high dose strategy? Do we know that?
Yeah, I'm going to put you 1 second. The only thing I was going to add to, no, the only thing I was going to add, I completely agree with everything that Heidi just said. I can just tell you my own personal philosophy, regardless of where the relapse is or how extensive the relapse is to always start with a phase 2 study rather than a phase 1 study. So just to refresh your memory, a phase 2 study is looking at a drug where we already know what the dose is, we already know what the toxicity is, and the study is being done specifically to look at response. So that's a step further down the road of what we know about that particular drug versus a phase 1 study is a drug that we think might have some activity because perhaps there's been something shown in the laboratory that suggests that it might be an anti-neuroblastoma type of drug, but we don't yet know what the dose is that we should use. So whenever there's a relapse, regardless of how extensive the relapse is, one of the criteria that I also take into account is what's in the phase 2 model versus what's in the phase 1 model. And I reserve the phase 1 model for after we look at phase 2 drugs.
I think I absolutely agree. I think that phase 2 agents, I think, will be appropriate. But one step further, though, is how do you combine the phase 2 agent with, as you were saying, the known agents we have, whether this is high dose or low dose. And I think that all I can say is you have to base it on what happened in the past 10 years. A lot of this it's a learning curve. We don't know. I mean, the physicians might think they know. I certainly don't know what is the best way to do it. But we just learn it by empiric experience. We tried this in so many patients, or maybe only one patient, and it looks like it worked. Therefore, we will continue doing it. And until the point where we begin to believe that maybe it does work. So I think a lot has to do with what has happened in the past. So I think it'd be important, since parents, obviously many of this is not published most of the time, or the study is not completed yet, it's really, you have to trust the doctor. You talk to the doctor and they say, well, we've done this now, so many times and it has worked. Meaning that besides getting someone back in remission, you actually maintain someone in remission, at least in what % of the time. And remission, let's say, past 3 years from relapse. So this is the kind of thing that you probably want to hear, has anyone gone through this and it's alive today? And that is entirely, depending on where you go and depends on the regimen. So I think it's very empiric. What is the best thing? And we all rely on empiric experience. Although sometimes just a pure scientific rationale may be all you need. But even beyond that, I think we should have some empiric experience. What happened to the last 10 patients who were treated this way? What happened to them? I think that's a very valuable way both for parents as well as physicians to bring things into the phase 3 fashion, is that in the phase 2 setting, what kind of response we get and what kind of long-term results do we have in a patient group like this.
I wanted to make sure that I can correctly answer Mark's question, which was, if you have a single lesion, is there one therapy that is better than another therapy? And there is not one therapy that we know is better than another therapy in that particular setting. So if your doctor recommended one thing, or if I chose one thing for one patient and another thing for another patient, it's not because I really have a whole lot of ground to stand on. It's just mostly about what I know about how that patient's going to tolerate drugs, how that patient's responded in the past. But there's not a drug out there that we know with any certainty is going to be the proper drug for that setting of tumor relapse.
Thank you very much, Ollie. You still agree?
When you're testing a drug in the relapse setting, what do you consider success? Because myself as a parent, if you give my child an oral chemotherapy and he gets 6 months of even stable disease where it was at, or a little bit better quality of life, I mean, I consider that phenomenal success to have 6 months not hooked up to an IV, and so he progresses again. I still consider that drug was absolutely amazing. You got 6 months where we didn't have to be physically stuck in a hospital up to an IV. So what do you guys consider success?
Well, the goal of a clinical trial is not the same as the goal of the patient entering a clinical trial. As Sue pointed out, a phase 1 trial is a descriptive trial. It's trying to determine what is a safe dose. So we've succeeded if we've accurately captured the side effects that the drug has caused and found a dose that we think is going to be safe for further studies. And we certainly are gathering information about how the disease responds at different dose levels, but because people are getting different doses, we can't make really strong conclusions. In a phase 2 study, it just depends on whether we think the tumor is going to shrink with the chemotherapy or whether we think it's going to be stable. And we designed the study so that we have a certain percentage of patients, usually around 20 to 40% of patients, that are going to respond in some way that we think shows the disease is being affected by the tumor. So if at the end of the trial, we can clearly say one way or the other that the disease was affected or not affected based on how we designed and kind of what our question was to begin with, we consider that a success. But that's the scientific success. We probably consider a success similar to yours. A child has good quality of life with minimal side effects for an extended period of time. I think that's an absolute success for that child.
I think with all the new agents as well, it's becoming very difficult to figure out which one is going to particularly work. So I think one of the new trials that's going to be coming through is actually which looks at take a backbone as far as chemotherapy and have actually sort of 3 arms, if you would, as far as different sort of biological agents. And it's going to be sort of a pick the winner strategy to see which one of those particular drugs added to a sort of known chemotherapy regimen is more effective. So that's what we're hoping to see in the future, to figure out how do we pick out those drugs, because many of the targeted agents that we're looking at now by themselves probably don't have a lot of efficacies as single agents, and that's one of the difficulties we're running into. With chemotherapy, it was, if chemotherapy worked by itself, we thought you could put it into therapy. But certainly these biological agents, they may work much better actually with chemotherapy, so that's one of the difficulties we're trying to figure out now.
So traditionally, you look at new drug development, 10 to 20% overall, what we call major response rate, either CR or PR, is regarded as something worth pursuing. And Patty can correct me if I'm wrong. But I think that with the new agents, especially as Darryl was saying, it's probably been likely going to work by itself. I think there's probably be very little activity in that kind of CR, PR, meaning a complete remission or partial remission type of response in this relapse setting. So I think that actually the demand is a little lower now. In fact, in many of the clinical trials, people are not even looking at response. They're looking at biological correlates to show that when a drug is given to a patient, it has an effect on the white cell, not on the tumor, but on some what they call surrogate marker, and that is defined as a success. So as far as anti-tumor, how this drug is actually killing tumor cells, it's even further away. So I think that if you be pragmatic about it, the best thing still is still looking at how much response do you get from a drug. And I think if you can get some major responses, I think that's a drug you should move forward to. And as to picking the winner, it depends on who the competitors are. If I go to the Olympics and everyone I run with is a handicapped person, I'm sure I'll be the champion. But the point being is that in drugs, the same thing happens. If all you have available is really not that great, so even a stable disease is better than nothing. So I think it depends on what your competing drugs are on the market and available at this point in time. Maybe tomorrow or a year from now, there'll be a different horizon. There'll be a lot more drugs available. Then you'll be competing truly this time for those that are winners. They're really home runs. But today, we just don't have that many that are that dramatic in terms of overall response.
How important is the time that relapse is catched for the prognosis of the disease? And if it is important, then how frequent must the patient be screened?
think that we've brought up the concept that if you find a tumor when it's a single lesion or a single site, that maybe you have a better chance of getting the disease under control. My experience has not necessarily been that that's the case. My experience has been that we're able to give the kid more therapies in large part because we have more time where we know the disease has relapsed. I don't know that there's anything clear to say that if we find a relapse when there's a single lesion versus when there's 10 lesions, that we have a better chance of getting the disease back under control. The disease will get us back under control with treatment or it won't. And so I think finding tumors early on has benefit though in that it does allow us to try therapies that are less toxic because we're not racing against what the disease is doing to the body. And it allows us time to work with the family and help them choose what they think is the right types of treatments. It gives a little bit more leeway in lifetime. But I don't think finding it early necessarily means that you're going to have a better outcome. We do have sort of scheduled imaging studies that we follow on the COG studies more frequently after initial therapy and then spreading out over time.
And I think there's one point is it may not be that you're catching it earlier. I think if you see a single lesion, it may be biologically that's different from the patient who 3 months ago the scans were negative and now you actually have 10, 15 different lesions. The biology and the behavior of that particular neuroblastoma may be different. So it's not so much necessarily always catching it earlier. It's maybe that patient's neuroblastoma just took off. And it wasn't that you missed the scans from three months ago. I think that's, and it may make it more difficult to treat. It may not.
It's obviously a very important question. How we follow patients in neuroblastoma? Are we doing enough testing or are we overdoing testing? Many times we're getting tests every 3 months, every 6 months. And we obviously, with the MIBGs that we're doing today with 123, we are able to pick up relapses even before the patient is symptomatic. And oftentimes it's a shock to the parents to say, how come my child is absolutely feeling fine? How could you find anything on the scans? But that's what we're seeing now, what we call asymptomatic relapse. In a study that Dr. Kushner summarized, he presented in some of these meetings, essentially what it is just looking back at our patient population, among patients who have asymptomatic relapse, those who were detected by MIBG 123 does seem to have a longer survival, median survival, meaning that if you pick up the tumor earlier, they seem to have a longer survival, counting from the time of relapse or counting from the time of diagnosis. So now there's a confounding factor, meaning that it could be just a totally different disease anyway. But at least in a retrospective study, it does look like that screening using MIBG scans and maybe similar studies could pick up early relapse that may have an impact. I think you need a randomized study, prospective trial to actually prove that. But at least in a retrospective analysis, I think it's suggestive that it may have an impact, may have an association at least with long survival.
We're still waiting for the 4B to tell us how to cure relapse disease. So we'll end the session after you tell us how to cure. Finally, the two things if you wouldn't mind commenting on. One, obviously the disease of relapse is not necessarily the same disease and diagnosis. And there's been talk within the neuroblastoma community regarding taking samples of disease, growing them out, and testing them against different drugs for treatment, to be able to tailor your treatment of disease. I'm curious if you could comment on the use of that and how that's going to be used in the future, if you see that being an integral part of treatment in the future. And then another comment. My other question you real quickly is to comment on after that is treatment of second remission. Assuming all goes well, they relapse and you go to the second remission. Is your approach to treatment treatment of second relapse, treatment of second remission different than treatment of first remission, just to comment on that for those who will be face that situation.
David, to answer your question about the culture, I think that it's an important aspect that we need to really do more of. And just on the phone with Mike Hogarty yesterday, actually talking about amending the COG neuroblastoma protocol to bring in all the relapse specimens. We're going to put them into non-skid xenografts, into special mice, into cell culture, and into special culture conditions. And are very uniform techniques to make sure that what we got from the patient is that sample. And that's one of the problems that we have, the 15 or 20 different labs doing it, is that we're getting some samples from them and testing them by molecular techniques, and we're finding that the cells they think they're growing up from that patient were actually cells that came from a cell line that's been around for years. So we have to be cautious about this. I don't think it's ready for prime time for individual patients, but I would encourage everybody to make sure that samples come into laboratories that are doing these kind of studies. From my perspective, preferably the COG lab, because that way we have all of the data in one place and we can share it there.
So I'd like to address David's first question from a clinical perspective. Even if we were able to type the patient's tumor and look and see what was upregulated and downregulated, that does not mean that that drug is going to have benefit for the child. So I think you have to use that kind of information with extreme caution. When scientific discoveries or things that are taken in one context are applied in another context, that can actually have negative impacts. You can be receiving a drug that may be dangerous in another setting. And so I think there's got to be a lot of steps between taking the profile of a tumor and what drugs might affect that tumor and interpreting that for any particular patient. So that was number one. And number two was the question about relapse or second remission. And we were kind of talking about this morning too, and the concept of considering relapse neuroblastoma as a chronic disease. And I think even when I have children whose tumors are no longer evident after relapse, the parents have to do a lot of jumping up and down and begging before I consider actually stopping therapy. Now, that doesn't mean I keep them on intensive therapy like tococyclo or something that needs to be life-altering, but certainly on some sort of oral regimen or something that seems to kind of keep the disease in check. And I think that's, do I have a lot of evidence to make that assumption? I don't really, except from the leukemia studies where they do that for 2.5 years and are able to kind of keep the disease a little bit more at bay. But I'm always frightened to stop therapy in a relapse setting. I think you know, if you're honest on a treatment that has good quality of life issues, that's not affecting the toxicities are reasonable. The question is then why stop? You know, if the risk is that the tumor can come back.
I just wanted to add one more thing to the cell line culture issue, and that is that, again, as we are looking at more drugs that perhaps work in different mechanisms other than just the way a chemotherapeutic drug might work, for example, the anti-angiogenic therapies that Daryl's working on, that is really not something that you can test in a cell culture per se. So that there are some things that you can learn from cell culture in terms of sensitivity to drugs or other drugs that this really isn't a great way of analyzing that. Some other types of studies need to be done. And there's always situations where you do a really good job in terms of curing the mouse, but that may or may not end up being as effective in the patient. So all of it is very good suggestive information, but you don't know for sure whether the laboratory will necessarily always translate into what we're going to see in the patient.
Yeah, two more questions. The young lady down there and then Brian.
Can I, what, question?
Oh,
Again, the chromogenic assay. It's an assay that's been around now for 20 years. When I was starting out, I was excited by it also. But after trying that and using it as a piece of information for treating patients, I was much more cautious because every single time I tried it, I actually did not agree. The drug that worked in the test tube was not benefiting the patient. So we're becoming much more cautious in terms of taking the data directly and they're using it as a treatment decision. But I think that obviously this is, again, a moving target. I think the tests are doing much more sophisticated now. I think a lot of things have improved. I think it will probably become a useful adjunct for the oncologist someday, and maybe the time is not now yet.
I think you just touched on it a bit, but I want to make clarification here for myself. I'm getting the perception that all we can do is get the disease under control and that's it. Is this just in a relapse setting that we're speaking of?
Yes.
And not up front?
There are children that are cured.
There are children that are cured up front where they go on to be cured and never have the disease come back. But in the setting of relapse, it is very, very difficult to cure a patient. And we see patients who do well for long periods of time, and maybe we will eventually see some patients truly have long-term, event-free survival. But most of us feel that we're dealing with a more chronic disease and trying to keep things under control when the patient relapses. That's not the case up front. Up front, we are curing patients.
In a small subset, I have to say, small subset, if someone did achieve a second remission, and by that I mean a complete remission of EGPR, at least in our data, those treated with antibodies to the GM-CSF, there's about 20% long-term disease-free survivors. Now, it could be just a patient selection. Again, you have to be very careful in terms of what are these patients. They're stage 4s, they obviously are high risk, but there may be some difference about the biology of the tumor. It could be just a totally, patient selection. So again, it's a small subset. As Sue was saying, a subset of patients does seem to be able to make it for a long time, long-term disease survival past the first 3 or 5 years. So I think that there's still a subset that we need to define. We need to find out a little bit more about maybe genetic studies, molecular studies, as to why they do better. And maybe that the way to really help us choose therapy in the future, and maybe even help the other group that didn't respond.
In relation to older children, Dr. Chung, you had made the analogy of the seed and the soil. What do you think specifically is changing in the older children? Is it genetics?
That's a very hard question. I don't know. I think for those of us who's been working in neuroblastoma, it's fascinating in some sense from a scientific way. Why is it so different in an adult versus a child? They look identical under the microscope. They also pick up MIBG. They grow as a mass. They go into the bone marrow. But in a child, it's so different when it's in an adult. The same thing you can say about infants. Infant neuroblastoma is so different from a child. Infant neuroblastoma is curable in 80% and more of the time without transplant, with just chemotherapy alone. So I think age is a very important factor. And we have to understand that somehow, someday. And I think all the research that's ongoing now hopefully will answer that question. And this is a $1 million question.
And I was just going to say, in my opinion, I really think age is a surrogate for the biology of the tumor. And clearly the biology of a tumor in a child who's less than 18 months, the actual tumor cell is different than the tumor in a child who's a toddler, and that's different than the tumor in a child, a young adult who's 17 or 18 or 19 years old. So I really think that we don't quite have the tools yet to be able to understand all the genetic variations. I think that it's just around the corner with all the genome-wide array studies that are being done and the epigenomic studies that are being done. And right now our surrogate for being able to try to figure out how an individual patient will respond, one of the factors that we can look at is age. But I really think that is a surrogate for the biology of the tumor.
I think definitely the age defines biology up front, but I think there is going to be maybe one more, and that is that the seed remains the same, but the soil is changing. And the fact that if we can keep the neuroblastoma going for 5 years, this tumor will probably not relapse for another 5 years. And because the soil has changed, the fact that this tumor now is sitting in an older child or sitting in a young adult, that tumor, for whatever reason it is slower in growth and slower to recur. So this is sort of a fascinating biology that we begin to appreciate because we're seeing a lot more chronic, what we call chronic neuroblastoma, but these are all also older children now. They actually got bigger, older now, but the disease has also changed.